So now we're going to be talking about glycolysis. Now what glycolysis means is if you break the two words up, it's glyco and lysis, which means the breaking down of sugar. So over here we're going to start out with glucose. Glucose will then go through a whole chain of enzymes, but the two main important ones is, the first one is hexokinase, which what it's going to do is it's going to take an ATP, turn it into ADP, and shove it onto the end of glucose. It's going to go through another couple enzymes, and then it's going to hit another enzyme called phosphofructokinase, which will do the exact same thing. It will take ATP, and it will turn it into an ADP, and then it will also shove a phosphate onto the other end of glucose. So now we have a glucose molecule with a phosphate on its 1 and 6 end, okay? So then it's going to go through a few more enzymes, which is actually going to then cleave that glucose molecule into two separate parts. We're going to end up with two PGALs, or in other words, a phosphoglyceraldehyde. So from here, phosphoglyceraldehyde is going to go through a whole chain of other enzymes. Now, as it's up here, what we're trying to do for this glycolysis is to create energy, which up here we've actually destroyed it. We turned ATP, which is usable energy, into ADP, which is not what we want to do. But this process will create energy. Now this process will happen twice because there's two PGALs. What we're going to make in this process is we're going to end up making an NADH, an ATP, and another ATP. But since this happened twice, we're going to end up having two NADHs, two ATPs, and then two more ATPs, for a net total of 4 ATPs because of the loss from up here, and 2 NADHs. The end result of all of this is to make pyruvate. Due to the fact that it happens twice, there will be 2 pyruvates. So our total creation is 2 ATPs, 2 NADHs, and 2 pyruvates in glycolysis.
Now we would talk about the TCA or Krebs cycle. After pyruvate has been made in the intramembrane space, it enters the matrix of the mitochondria via the pyruvate entry complex, or the pyruvate dehydrogenase complex. After it enters, pyruvate becomes a carbon dioxide molecule, an NADH molecule, and an acetyl-CoA molecule. Acetyl-CoA then converts oxaloacetate into citrate. It's where then it goes through a bunch of enzymes which creates numerous NADH, GTP, and FADH2. And after it goes through this cycle, our total creations are 10 NADH, 2 FADH2, and 2 GDP.
Alright, the last thing we're going to talk about is the electron transport chain. So, first thing that's going to happen is, over here in complex number 1, what's going to happen is the NADH that was made from before is going to enter it. Or not enter it, but donate its electrons. And it's going to turn it back into NAD+. What this is going to do is it's going to cause the H that was attached to the NAD+, or the NADH, and shoot out of from the inside of the matrix into the intermembrane space. So it's going to take an H+ and shoot it out, which is going to donate then 2 electrons to complex 1. Now, what these 2 electrons are going to do is they're going to shoot down this entire chain of electrons. The reason being is over here, there's an O2 molecule, an oxygen molecule, which is very, very, very electronegative, and it wants to collect all of the electrons. Like I said, the electrons are then going to shoot through every single complex until it reaches the oxygen. The oxygen will then splice and become 1/2 H2O. Now over here in complex number 2, NADH won't attach to it, but the other electron shuttle that we made earlier, FADH2, will. So FADH2 will connect onto complex number 2 and turn it into FADH. This will also shoot hydrogens out into the intermembrane space and then shoot its electrons down the chain. Now, what's going to happen is, as every single time an NADH or an FADH2 attaches and donates its electrons, H pluses are going to keep constantly going out of the matrix into the inner membrane space. What we have over here is something called ATP synthase. Now, what this is going to do is it's going to take an inorganic phosphate and an ADP and convert it into an ATP. But in order for this to happen, it needs the entrance of H plus back into the matrix. Now, like I said over here, where it keeps shooting the H pluses out of the matrix into the inner membrane space, there's a high concentration of H pluses outside the matrix, causing an electronegative gradient. Electrochemical gradient, I'm sorry. Now what's going to happen is, based off of diffusion, these H pluses are going to want to come back into the matrix, which is what's going to happen. They're going to shoot into the matrix, and it's going to turn, this ATP synthase is going to turn it and create ATP from an inorganic phosphate and an adenosine diphosphate. Now, this whole process of creating ATP through the electron transport chain is called oxidative phosphorylation. This is because, right here, the oxygen molecule is very electronegative and wants to collect all the electrons, causing the turning of... This whole process is called oxidative phosphorylation because of the oxygen molecule right here that wants to collect all of the electrons and start the entire process. The end result of oxidative phosphorylation in the electron transport chain will create between 30 to 36 ATP molecules. Now, combine that with what happened in glycolysis, and we end up with between 32 and 38 ATP molecules. After discussing everything that we've done and what happens in the process of cell respiration, this is how energy is created. By taking a simple glucose molecule, breaking it down through a lot of enzymes and creating ATP, or in other words, adenosine triphosphate, the usable chemical energy of our body.